Welcome to Mythosium, the channel where we take complex scientific ideas and make them feel simple, intuitive, and surprisingly human. Today we're exploring something that sounds almost mythical, almost too strange to be real: the connection between horses and snakebite survival. It turns out that one of the most effective tools for treating venomous snakebites doesn't come from a lab, a synthetic formula, or a high-tech device. It comes from biology, specifically the remarkable immune system of the horse. At first, the idea seems unusual. Why would horses be involved in producing antidotes for some of the most dangerous venoms on Earth? Why would this particular animal play such a fundamental role in a medical procedure that saves thousands of lives every year? And what does a horse have that makes it uniquely suited to helping humans survive encounters with venomous snakes? To understand the answer, it helps to first look at what venom actually does. Snake venom is not a single substance but a mixture of proteins, enzymes, and toxins that act together like a biochemical weapon. Some venoms attack the nervous system, shutting down communication between nerves and muscles. Others target blood clotting, causing uncontrolled bleeding or, paradoxically, dangerous clot formation. Some destroy tissue directly, breaking down cell membranes or digesting structural proteins. And many venoms combine several of these effects at once. Because venom is a cocktail of diverse molecules, the immune system must recognize and neutralize not just one threat, but many. In humans, our immune response is powerful, but it takes time to build up defenses. Venom acts far too quickly. Even if the human immune system could eventually produce effective antibodies, the venom would cause severe damage long before that response became useful. That's why humans can't rely on natural immunity to fight venom. We simply don't have enough time. This is where horses come in. Horses have large bodies, robust immune systems, and the ability to generate significant quantities of antibodies. When exposed to controlled non-lethal doses of snake venom, a horse's immune system reacts by producing antibodies tailored to the venom's molecules. These antibodies circulate in the horse's bloodstream, seeking out the components of venom and neutralizing them. Over time, and through carefully managed repeated exposures, the horse builds an immune defense strong enough to counteract even potent venoms. The key idea is that the horse is not harmed during this process. The initial dose is far too small to be dangerous, and each subsequent exposure only occurs after the immune system has strengthened. The horse's body adapts, responding like a biological factory for highly specialized antibodies. And because a horse's bloodstream is large, it can produce these antibodies in significant quantities, far more than a smaller animal could generate. Once the horse's immune system has built a strong defense, trained professionals collect a small amount of its blood. From that blood, the plasma is separated, and within that plasma are the antibodies, the very molecules that can neutralize venom. But these antibodies are not used in their raw form. They must be processed, purified, and refined to remove proteins that could cause human immune reactions. What remains after this purification is what we call antivenom, a concentrated preparation of antibodies designed to seek out and bind to venom molecules inside the human body. When antivenom enters a human bloodstream, it acts almost like a guided system. The antibodies circulate until they encounter the venom, latch onto the harmful molecules, and block them from interacting with the body. This process doesn't reverse damage that has already occurred, but it can stop the venom from causing more harm. The faster antivenom is given, the more effective it is, because it prevents venom from spreading further. This raises a fascinating question. Why horses specifically? Why not use another animal? The answer lies in a combination of biology and practicality. Horses produce large volumes of blood and antibodies, tolerate controlled exposures well, and develop strong, lasting immune responses. Their immune systems can handle a wide range of venom types, including neurotoxic, hemotoxic, and cytotoxic components. And because of their size, one horse can produce enough antibodies to help many people. But horses aren't the only factor in this process. The science behind antivenom also depends on understanding how antibodies recognize their targets. An antibody is shaped in a way that allows it to fit onto a specific molecule, almost like a key fitting into a lock. Snake venom molecules, despite their harmful effects, have structural features that the immune system can recognize. Once the horse's immune system identifies these features, it creates antibodies that bind tightly to them. This binding prevents venom proteins from interacting with human cells. In many ways, the antibodies act as molecular shields. This insight, that biology can create an antidote by studying and countering the structure of venom, reveals something remarkable about how life adapts. Venom evolved as a tool for predation and defense, designed to disrupt normal biological processes, but the immune system, with enough time and exposure, can respond by crafting precise molecular tools to counter those toxins. Antivenom is therefore a product of evolution and engineering working together. Evolution creates the toxins, and immunology creates the defense. Another important part of this story is the diversity of venoms. Not all venomous snakes produce the same toxins. Some venoms are more focused on affecting nerves, others on blood, and others on tissue destruction. In some parts of the world, venom composition can even vary between populations of the same species. This variability makes the production of an effective antivenom challenging. A horse needed to generate antibodies that recognize a broad range of venom components, which means exposure must be designed carefully to trigger a wide immune response. Professionals often mix venoms from multiple snake species to create polyvalent antivenom, capable of neutralizing different types of bites. This complexity also explains why antivenom isn't universal. Antibodies that work well on one type of venom may do little against another. The immune system learns to recognize specific molecular shapes, not general categories. That's why antivenom use must be matched to the type of snakebite whenever possible. A mismatch means the antibodies won't bind effectively to the venom molecules, leaving the patient unprotected. Given all this complexity, it's natural to wonder whether antivenom could ever be created without using animals. Researchers have explored synthetic antibodies, engineered immune systems, and recombinant technologies. These methods hold promise, but none have yet matched the efficiency, diversity, and adaptability of an actual biological immune response. A living animal can generate a wide array of antibodies in parallel, something that is challenging to reproduce artificially. Horses are not just producing molecules. They're producing an entire spectrum of immune responses tuned to the structure of venom. Until synthetic methods advance further, horses remain an essential part of this medical process. Beyond the biological details, the story of horses and antivenom reveals deeper themes about how humans interact with nature. Snake venom exists because of evolutionary pressures that shaped predators and prey over millions of years. The immune system evolved as a defense mechanism against pathogens and foreign proteins. Horses evolved strong immune responses to survive in environments filled with threats. Humans discovered a way to bring these elements together, using one form of biological adaptation to counter another. It's a reminder that solutions to deadly challenges often come not from rejecting nature, but from understanding it. Another fascinating aspect is how the effectiveness of antivenom depends on timing. Venom moves quickly through the body, spreading through tissue, entering the bloodstream, and interacting with cells. Antivenom can only neutralize venom it can reach. If venom has already bound to receptors or begun damaging tissues, antivenom cannot reverse that damage. This highlights the importance of rapid treatment. The sooner antivenom is given, the more venom it can neutralize before it binds to critical systems. This interaction between venom and antivenom makes snakebite treatment both a medical and logistical challenge. It requires quick access to medical care, proper diagnosis, and knowledge of which antivenom to use. In many regions, this process is complicated by distance, climate, availability of medical facilities, and variable venom compositions. Even though the biological mechanism of antivenom is well understood, delivering it effectively requires coordination, infrastructure, and preparation. But while antivenom is incredibly effective, it's not without risks. Even after purification, the antibodies produced by horses can sometimes trigger immune reactions in humans. These reactions occur because the antibodies, while helpful, are still recognized as foreign proteins by the human immune system. Modern production techniques aim to reduce these reactions by breaking antibodies into smaller fragments or removing parts that cause strong immune responses. These refined products are safer, more predictable, and better tolerated. The science continues to advance. New techniques in protein engineering, genetic sequencing, and immunology offer the potential to build antivenoms with greater specificity and fewer side effects. Researchers are developing ways to analyze venom structure at the molecular level, allowing for more targeted antibody design. There's also interest in creating an antivenoms tailored to the venoms of specific regions, reflecting the variability found in snake populations. As technology progresses, the process may become more efficient, more precise, and less dependent on large-scale animal involvement. Yet for now, the horse remains a remarkable partner in human medicine. Its immune system provides the foundation for antivenom production. Its biology bridges the gap between venomous threats and life-saving treatments. And its contribution illustrates something profound. Solutions to complex biological challenges often come from understanding the strength of different organisms and aligning those strengths with human needs. 手咬伤是人类面临的最古老的危险之一。 但现代的应对方法建立在深刻的洞察、科学创新以及对自然免疫系统的巧妙运用之上。人类与马匹合作生产抗蛇毒血清，是应用生物学最优雅的典范之一。这是一种生物体间的协作，利用一方的优势来保护另一方免受伤害。在 Mythosium， 我们热衷于探索这样的理念，因为它们展示了生命之间是何等的紧密相连。它们揭示了科学认知如何将古老的危险转变为可控的风险，以及生物学如何持续为那些曾看似无法克服的难题提供解决方案。If you enjoy uncovering the hidden mechanisms that shape our world and discovering how life's complexities can be turned into powerful tools, make sure to subscribe. Every week we dive into mysteries like this, bringing clarity to concepts that shape science, medicine, and human survival. Thanks for watching and stay curious.